Okay, so we are going to start now with Javier Vázquez, who is an anesthetist in the Nutri Neuro Lab in Bordeaux University, France, and he will be talking about ventral tegmental area projecting dopaminergic neurons as a neural substrate for flexible adaptation of goal-directed behaviors. So it's all yours, Javier. You need to unmute.
Let's do it. Thank you, Adeel. Hi, everyone. So, my name is Javier Vázquez, and wait a second. And I'm gonna be sharing with you the set of data that I got during my internship at the Bordeaux Neuro Campus as a graduate student, and under the supervision of Andrea Contini and Pierre Trifiliev. They are under the direction of Pierre Trifiliev. The resulting project is called Ventral Tegmental Area projecting dopaminergic neurons as a neural substrate for flexible adaptation of goal-directed behaviors. So goal-directed behaviors are a very important component of cognitive function based on the association between an action and an expected outcome. So if, for example, after a long day of work, I feel tense, which is an aversive state, my goal is to feel better, less tense, a more rewarding and pleasurable state. One possible action that I can perform is, for example, to smoke a cigarette. One important characteristic of goal-directed behaviors, nonetheless, is that they are flexible and can be adjusted when the context is changing. So let's say that now the context is really changing because I'm realizing when I'm reading this paper that my cigarette is really bad for my health. In fact, I realized that one single cigarette, as you can see, can reduce my life by 11 minutes. Then, what can I do? In a flexible manner, I can adapt my behavior, quit smoking, and, for example, learn mindfulness techniques to feel better, less tense, and live a healthier and longer life. Nonetheless, we know that patients suffering drug addiction persist in consuming the drug, although they know the negative consequences of drug use, showing deficits in this ability to flexibly adapt goal-directed behaviors. At the neurobiological level, alterations of cognitive functions in those pathologies are believed to rely on perturbations of the prefrontal cortex activity, including for sure the prefrontal cortex, and in particular, and that's very important, of dopamine transmission within this structure. However, preclinical data regarding the implication of dopamine signaling in executive control remain controversial. So, on the basis of this discrepancy, our goal during this project was to assess the implication of dopamine neurons of the VTA, the ventral tegmental area, during reversal learning and sensory-specific outcome evaluation task, since we know that the VTA is the main source of dopamine reaching the medial prefrontal cortex through the mesocortical pathway. Our hypothesis was then that an inhibition of VTA dopamine transmission would impair the flexible expression of goal-directed actions. To do so, we used a chemogenetic approach to selectively and reversibly inhibit dopamine neurons of the VTA. First of all, we used a DAT3 line in which the Cre recombinase was expressed in dopaminergic neurons. Then we injected an enterograde virus into the VTA carrying the Cre-dependent inhibitory DREF. And finally, we injected CNO peripherally and just before the behavioral test. What CNO did was to activate specifically the DREF, and the DREF itself inhibit the VTA dopaminergic projections. mainly onto the medial prefrontal cortex and the nucleus accumbens. So regarding the experimental procedure, first of all, during our instrumental training, the animals learned to perform an action that in this case was to press a lever to get a reward in a milk pellet, let's say, for example. And during the same sessions, animals learned to perform another action to press the other lever to get a different reward, a grain pellet, with the same calories. Here I'm showing you the results of instrumental training. As we expected, both groups increased. You can see the number of lever presses per minute along fixed and random ratio sessions, with no significant differences between groups and without session per group interaction. Importantly, mice were kept under caloric restriction and maintained at around 80% of their initial ad libitum weight. During the outcome evaluation task, we tested the animal's ability to flexibly use the association between the action of lever pressing and the outcome of getting a reward when the value of the reward changed. How did we change the value of the reward? So we did that by inducing satiety. So first of all, animals were fed ad libitum with one of the two rewards, the milk or the grain pellet, and this fact decreased the value of the reward. And at the end of this phase, CNO was injected. Right after, during the extinction test, both levers were presented, but lever pressing was unrewarded. So here we recorded the number of lever presses along this session. Here I'm showing you the results of this extinction test. We expected that the control mice, the cream minus group, the black one here, would reduce the lever pressing regarding the devalued reward, as they did, as you can see, adapting their behavior in a flexible manner. But importantly, although we predicted that the DREAD group, the CRE plus group, would press similarly on both levers, showing this impaired ability to adapt their behavior in a flexible manner, what we found was that the group expressed outcome evaluation during the extinction test, showing a higher amount, as you can see, of lever presses performed on the lever associated with the reward that was not devalued. Nonetheless, we found an effect of group, suggesting the differences in the total amount of lever presses performed by both groups of animals during the test. Here, I'm showing you the total amount of lever presses performed during the extinction test per group. As you can see, we found that the number of lever presses performed by the CRE plus group during the extinction test was significantly lower than that of control mice. So this data suggests that the BTA, dopaminergic transmission, is not necessary, as we saw, for the expression of goal-directed behaviors in the outcome evaluation paradigm, but it confirms its important role for maintenance of incentive processes at the motivational level. We then tested the effect of BTA dopaminergic inhibition during a reversal learning paradigm. Why? So we wanted to go further and test the animal's ability to flexibly respond to changes in the environment, since this ability is critical for adaptive behaviors. So following the last outcome evaluation testing that we just saw, the same cohort of mice underwent 4 sessions of instrumental reversal learning. Rewards delivered after lever pressing were now reversed across the right and the left lever. And importantly, this time, mice were injected with CNO each day, 30 minutes before the beginning of the sessions. Here, I'm showing you the results of the instrumental reversal learning procedure. As you can see, lever pressing performance per minute increased across reversal learning training days and did not differ between groups. Another choice test was run after this reversal learning phase. The procedure, I'm not going to get into the details because was identical to that run following first and second evaluation. But importantly now, no CNO was injected during this phase. And here, and this is a very important slide, I'm showing you the results of the extinction test following this reversal learning training. The control group responded significantly more on the valued level compared to the valued one. While this time, the CRE+ group equally similarly responded on both levels. So this was our very important finding, and this is suggesting that an inhibition of dopaminergic transmission during the acquisition of this reversal learning really impairs the expression of behavioral flexibility after the outcome evaluation paradigm. Importantly, as I am showing you here, this time no differences in the total amount of lever presses performed by the two groups of animals were found at the motivational level. So in conclusion, what we found is that, first of all, the BTA dopaminergic transmission has a critical role for maintenance of incentive processes, as it has for sure been demonstrated before. And we found finally that an inhibition of BTA dopaminergic neurons perturbs the expression of the outcome evaluation of this flexible manner to adapt to the new situation, but only following a reversal of the outcome evaluation contingencies, showing its specific role to encode new and reverse instrumental learning contingencies. So this is all. Thank you very much, and I will be more than happy to try at least to answer your questions. Thank you.
Thanks a lot, Javier. Is there any? So, are there any questions for him? It was a very great talk, by the way. It was very fun. Any questions? Okay, so probably I can ask. I'm not really animal researcher. I work mostly with humans, so pardon my ignorance a bit here. So these are the results that you're showing me are all behavioral results, right? So you have given pharmacological intervention here, but so what's the plan? So do we want to know what's actually neural mechanisms behind reversal learning and devaluation, or is something that
No, I thank you for the question. Yeah, no, we really want it in this world, especially with what is going on, is we really need to know the neural mechanisms for adapt as fast as we can to a new situation, no? And the reversal learning paradigm is really useful when working with animals for exactly doing that, for exactly checking that, no? Because what we are doing is reverse the rewards in a simple situation, and we can see if by shutting down this area of the brain, animals can adapt or not. So that was the point, yeah.
Okay, that makes sense. So there's a question coming for you from Mehmet Fateh. So would you like to ask the question yourself, Mehmet? I can allow you to speak if you like.
I think it's better if you ask because my voice is not good.
Not a problem. I think it's good enough to ask, but I can do it. So the outcome. So the question is this: that outcome devaluation is also some form of motivational modulation. So why VTA is involved in the later but not former? Maybe you can explain the difference between devaluation versus motivation.
Yeah, for sure. I would say that, yeah, when you are evaluating, you really have to be motivated for the evaluate in this survivor mode on, no? So yeah, that's a great question, and as we saw, no, in our data, motivation is really important when, and it's using a different pathway, a different neural mechanism, possibly. What we did was to shut down, and I don't want to be speaking longer. We shut down the whole dopaminergic system. So we really don't know if motivation is more involved with the nucleus accumbens following our study, or with the prefrontal cortex. But for sure, there is a different neural pathway for motivation, and that's what we saw. No? That you can devalue, but and you can be not that motivated, but by checking with the lever pressing. Yeah.
Okay, thank you, Javier. So thanks a lot again. It was a great talk. We really enjoyed. So could you please stop sharing your screen now so that Lindsay can